Hello, everybody. It's Miss Peachy again from your WCA Biology A class. And I'm looking here at Unit 4 in your biology curriculum. And this is your first portfolio for the semester. You only have two portfolios this semester. And each one of them you will be doing kind of more legitimate science type of portfolio. You're going to be writing lab reports. Now, I know if you had physical science with me last year, you kind of started off writing some lab reports as well, so this shouldn't be too unfamiliar with you. We're going to just kind of, I guess, modify. I wouldn't say modify, but the structure of these is going to be more refined, more kind of representative of what an actual scientific research paper would be like. But it's also guided, so relax. I have your back on this one. All right, so in this portfolio or in this unit here, we are talking about feedback and feedback mechanisms. Homeostasis. So maintaining homeostasis. How does your body maintain homeostasis? Well, first of all, what is homeostasis? Homeostasis is basically a biological state of equilibrium. There are certain things in your body that have to stay in a state of kind of control. And if they go out of that state of equilibrium, then disease happens and ultimately death can result if homeostasis is not maintained. So your body has plenty of different mechanisms that automatically happen when you are challenged with a certain stimuli, environmental stimuli, or internal stimuli that put your body out of a state of homeostasis in order to get you back into homeostasis. So to keep you alive, essentially. So, as you can see from the picture here, this dog is panting. It's very hot outside, or maybe the dog was running around and its internal body temperature has gone up. And every organism has like an internal body temperature that is the most stable for them. In order to reduce that internal body temperature, the dog pants. The surface area of the tongue provides a great area for heat exchange with the environment and is able to cool down the dog's blood and bring down the internal body temperature to maintain homeostasis. So, in order to maintain homeostasis, we have a variety of different feedback loops that occur within our body. It's called, they're feedback mechanisms. And either the feedback system is negative or positive, and that negative doesn't refer to it being bad, and that positive doesn't refer to it being good. It depends upon kind of what's happening. Um, there's always some sort of stimulus, an action that causes a change. And with a negative feedback system, the system moves away from that stimulus. The dog is hot, it wants to be cooler. So it's moving away from the hot to being cool. That goes through a negative feedback system. For a positive feedback system, the stimulus actually, the feedback system actually moves the body towards the stimulus to become kind of more amplified, and eventually resolution happens when the stimulus itself is removed. So we're going to go through a couple of examples of negative and positive feedback systems here. So I'm going to go through a couple of these with you. The lesson gives similar ones and then one that's different. Like this second one that I'm going to give you is not mentioned in the lesson, but I think it's a really good example. So for our negative feedback system, the dog thing is a good example, but we don't pant generally when we're hot. If you do, you shouldn't. It's kind of strange. So don't do that. But when we are hot, we do sweat. So sweating is a way to control our internal body temperature as well. So here's our negative feedback. Now it's called a negative feedback system. You can also call it a negative feedback loop, but we'll just keep system because that's what the lesson is saying. Okay. So, for example, your body temperature goes up. So I'm just going to use an arrow to represent going up. Increase in body temperature. Okay, what happens with that? It results in I'm just going to actually outline this in orange because that's our actual mechanism. It results in sweating. So that's your kind of mechanism there to deal with being too hot. The sweating causes evaporative cooling of your body, right? So that leads to we're sort of writing at the bottom of the screen. It gets kind of difficult. And that leads to a decrease in body temperature. Oh, right? So resolved at that point. Now, if you get too cold, so your body temperature goes down, maybe it can be depressed too much. You start getting too cold, what's going to happen? Well, you start getting goosebumps and shivering. So that's another mechanism. So in this case, the stimulus is I'm too cold. So then the mechanism is goosebumps and shivering, and that is going to lead to an increase in body temperature. Okay, so you've got several different mechanisms depending on whether or not you're too hot or too cold, but the end result is to get you back into a state of homeostasis, an ideal body temperature for you. Whether that's, for humans, it's on average 98.6 degrees, but it varies from person to person. So it is kind of getting you back into that average body temperature to get you back into a state of homeostasis. So that's a negative feedback system because your end result is to get to an opposite state. Increase in body temperature, you want to get to the decrease. If you're too cold, you want to get to warmer. So it's the opposite of what you began with. Our positive feedback system is only resolved by removing the stimulus. So you can't resolve this by removing the stimulus. Essentially, the only way you can remove the stimulus is to just keep increasing and increasing and increasing the initial stimulus to be able to finally get it to go away. So, like I said, I'm going to use a different example. I'm going to use childbirth because that's a good example. So, in childbirth, childbirth is typically started through a release of a chemical, a hormone in your body called oxytocin. So your brain releases oxytocin. So oxytocin is a hormone that triggers contractions in the uterus. So what this does, it triggers, causes uterine contractions. Well, just so happens that if your uterus contracts, that is going to stimulate the release of oxytocin in the brain, which then stimulates more uterine contractions, which stimulates even more oxytocin to be released. And so basically, during the process of childbirth, the contractions just get more and more intense as time goes on until the baby is delivered. Once the baby is delivered, then the brain, the delivery of the baby slows down the uterine contractions, which slows down the release of oxytocin and eventually stops it altogether and the contractions stop. But the only way to kind of go back into a state of homeostasis here is for the um, the whole process to kind of be, I guess, the position of the baby initially is what probably triggers the brain releasing the oxytocin. And so being kind of delivered removes that initial stimulus. So then you no longer have contractions taking place. So that's an example of a positive feedback loop because one is actually causing the amplification of another. So in this lesson, we are going to be exploring specifically the first feedback mechanism. We're not going to be exploring childbirth in having you guys do any kind of labs with that. But we are in looking at the stimulus that causes body regulation of body temperature. So maintaining homeostasis in your body temperature. Talks about feedback mechanisms here. What are the kind of things that actually causes these changes, that sweating and goosebumps and shivering. They also talk about, I didn't mention, vasodilation and vasoconstriction. So your blood vessels will actually dilate and shrink up depending on whether or not you want to maintain heat or you want to lose heat. So vasodilation, increasing the diameter of the blood vessels, allows more heat to kind of escape through your vessels as well. But those are mechanisms. Those are physical changes that help to kind of resolve the situation. So what you're going to be doing is you are, I can get the head up here, going to be constructing a lab report where you're going to be testing doing some sort of a test for that first one, the biofeedback mechanism of maintaining kind of your homeostasis in your body. So I am going to, here we go. So I'm here on slide 9. If you're on slide 9, it'll all the way go down to the bottom. It'll give you kind of an example of what you might want to do here for your lab. Essentially, you're going to be writing a lab report while you're doing some sort of experimentation, testing some variables regarding maintaining homeostasis in the circulatory system. Now, I'm going to give you some examples because I don't want to just let you loose on this and you guys don't really have a great example of what you want to do. Okay. So first, let's talk about a couple of terms here. In any kind of scientific investigation, you have something you're testing. So generally, you start with a question that you're asking. And from there, you need to identify what your independent variable is going to be. So this is the thing you're actually testing or changing in the experiment. And then you're going to see what actually gets affected by you changing that particular thing. And the thing that actually is the results of your experiment, what you are testing for, is called your dependent variable. So independent variable, and I'm going to use my little typey tool for this one, I think. This is what you are the experimenter, right? Change or are manipulating during your experiment. This is what you're trying to see if there's any effect from this, okay? Dependent variable is the thing that responds to the independent variable. So a good example of this is if I want to see what the best fertilizer is going to be for my plants at home. I might buy two different kinds of fertilizer. Maybe I'll buy like the generic version and I'll buy Miracle Grow because I know Miracle Grow is good, but it's expensive. So maybe I want to be kind of cheap and buy the generic stuff, but I don't know if it's as good. So my independent variable is going to be my type of fertilizer, right? Whether I'm using regular or just or Miracle Grow or the regular cheap stuff. And then I'm going to see, can I test it to see if one's better than the other? I have to have some measure of how that is going to be tested. So maybe I'm going to see how like big my flowers are. So the size of my flowers is my dependent variable. So this is a thing that's going to be different from the Miracle Grow group to the generic group. And I can use that as a measure to determine if my independent variable actually has any kind of effect on the flower size, the dependent variable. So it's a measure essentially. So in your experiment, you have to kind of identify these as well. So I have a lab report template that I have made up for you. So you're going to be able to use this for this portfolio, and you can actually adopt this and use it for many different portfolios or lab reports in the future. So your first section of your lab report will be your introduction. In this section, you are going to explain the concept you're studying and how the lab that you are doing explores that concept. And then you're also going to include the question or problem you're trying to answer, write a hypothesis about it. So in this particular experiment, I have a couple of ideas for you. So let me put those on here. Okay. So the one that's given as an example, you can use that one if you want. In that example, you are going to see how the intensity of the exercise you do affects your heart rate. That's the example that they give you in the lesson. If I do low intensity, moderate intensity, or high intensity high intensity exercise for, let's say, 2 minutes, how does that affect what my heart rate, my active heart rate is going to be? So you can kind of imagine how you could devise an experiment to test that hypothesis. That's the one that's given in the lesson. Another one that I came up with is, how does the length of recovery time affect your recovery heart rate? So in this example, you would picture yourself doing, again, 3 different trials of exercise, all at the same intensity, though. You wouldn't change the intensity. You would do like 2 minutes of exercise, and then you would wait like 1 minute and then take your heart rate and see what it is after 1 minute. Repeat the exercise, do 2 minutes of exercise, wait 2 minutes and check what your heart rate is. And then again, repeat it and do 2 minutes or do 2 minutes of exercise again. And then wait 3 minutes and check what your heart rate is. So your recovery heart rate is basically how long does it take you to kind of get back down to just a regular resting heart rate. So again, you would do multiple trials of exercise and then wait increasing lengths of time before you took your pulse to see what your heart rate was at that time. Another one that I came up with is boys versus girls. Maybe you have a brother or a sister, and they would be willing to help you with this experiment. So, boys versus girls, is there a difference in the resting heart rate? So, your independent variable would be gender or sex, if you will, here, boys versus girls. And the dependent variable would be resting heart rate. Now, resting heart rate is your heart rate when you are at rest. Right now, it's probably not a good idea for me to test my resting heart rate because I'm not truly at rest. You would probably want to lay down and kind of relax for a good 15-20 minutes to be able to get a better measure of your resting heart rate. So if you're not really keen on doing one where you have to do exercise, then this might be a good one for you. Boys versus girls, resting heart rate. So again, you're checking the difference between boys and girls. Is there a difference when it comes to resting heart rate? Granted, you don't have a huge sample size here, but that's all right. You can do the same thing, kids versus adults. Maybe instead of a brother or sister, your parent or your grandparent can help you out here. Does age affect your resting heart rate, right? So there's a bunch of different things you could think about here for variables and how it might affect your resting heart rate. So those are a few different examples of things using the circulatory system. They kind of want to look at homeostasis of the circulatory system in this particular lab exercise. You can pick one of these four here. If you want to pick something completely different, just talk to me first and maybe kind of run it by me, and I can let you know if that's something that would work, or we can refine it together if need be, or just let you know it's okay. So just talk to me before you do that. So once you establish what it is you're going to do, then you can come to the introduction. And again, you're going to explain what concept you're studying. We are studying homeostasis and biofeedback, and feedback mechanisms, right? Feedback systems. So you want to talk a little bit in this section about what homeostasis is and what feedback mechanisms are, because that's kind of what we are studying. Then you're going to talk about how your problem, let's say, is I want to see if there is a relationship between how much or how hard I exercise and what my active heart rate is. And then you need to write a hypothesis. And so hypothesis, oftentimes when students are younger, we say a hypothesis is an if-then statement. But now that you're kind of a little bit older, we want to add a part to that. Your hypothesis should be an if-then-because statement. If I do this, then I think this will happen because, and this is a good way to include the information about feedback mechanisms here, right? So if I increase my intensity of exercise, then I think my active heart rate will do this, and this is the reason why I think that. So make sure you have a hypothesis in here, an if, then, because statement predicting what you think is going to happen. Generally, you should be making these before you do the experiment. So the idea is you're trying to figure out what you think is going to happen before you actually figure it out. Once you've completed the introduction for this, then it's on to the materials or the methods section. So the methods section includes several parts. The materials that you need to do your experiment, a step-by-step procedure of what you did, variables, here you're going to include that independent and dependent variable we talked about before, but also you should include something called constants. Constants are also sometimes called controlled variables, but these are things that you did the same in all of them. Okay, so for example, if we were doing the length of recovery time, if we did this one, what we would do the same is we would still be doing the same exercise after every time we tested the recovery period, right? We're not going to change the exercise. We're not going to change the length of the exercise that we did. We're going to continue to do the same exercise, right? So you have to make sure you do that as well. The intensity of the exercise, how does it affect your heart rate? Again, you want to make sure you do the same, like if you're doing, let's say, running in place, do running in place for all of them. Don't do running in place and then do burpees, and then, because you don't want to change the type of exercise. That's going to be something that might have an effect on the outcome. So you want to keep everything else the same except for the one thing that you are testing for. So those things are called your constants, the things that stay the same. So that's what goes here. In your variables, you got your independent variable, that's what you're testing, your dependent variable, that's how you measure what you're testing, and then your constants. The materials, well, again, if you're measuring like the intensity of your workout, maybe you just need like a stopwatch or something to measure your pulse. Or if you're kind of real, get some cool things at home. I have a little finger pulse oximeter that I got during COVID, which you could actually put on and measure the pulse itself. Or maybe have on your phone, there's an app that measures your pulse. I know some of them can do that as well. But you have to have some way to measure your pulse. At a minimum, you need a clock or a stopwatch to measure your pulse. You need probably, let me think. I mean, honestly, for that one, you might, if you're doing some sort of an equipment, let's say you're riding on a stationary bike or something, maybe you would need the stationary bike. So you just have to figure out what materials you need. And then the procedure. And in this case, the procedure is probably going to be very simple. But if you were trying to explain to somebody step by step what you did, you would list those steps there under the procedure. We talked about variables, and then under safety, any kind of safety concerns that you need to be aware of. Um, think about, if you feel dizzy, you need to make sure you sit down, or make sure you have a glass of water handy just in case the person feels not like too hot or something. Just there might be some safety concerns for your health in this situation. Next section is your results. In the results, you should have your data in tables and a graph. And the data in tables, so a data table. So you have to figure out kind of how to organize your data. Basically, I mean, it's going to depend on which type of experiment you did. But, oops, that's not what I wanted to do. I would go to Insert Table here. And that's if I'm doing boys versus girls and resting heart rate. Let me just think. I'm going to probably put 3 rows and maybe, okay. Maybe you have more than one person, which would be kind of cool. But if I just had 2 people, boy and my girl, and then I maybe will take just for 2 different examples just so I can get some data, right? So maybe in this situation, I would have us rest for 20 minutes and then measure our resting heart rate on one day. And then the next day, I would do the same thing. And then I would average those 2 together for an average column. So you'd have some sort of a data table here. You're also going to have a graph. How can I graph this? Well, if I have 2 numbers that are averages, I can make a bar graph showing this information. So in this, you would go Insert, Chart, and you can maybe select the bar graph here. And if you need some directions on how to do a graph or how to make a graph on Word, I can walk you through that as well. Just kind of give me send me an email. We can go through that in my live lesson room. Or if you'd rather make a bar graph by hand, that's fine too. What I would ask of you in that situation, if you're going to do a bar graph by hand, which some of you will feel much more comfortable doing, then what I would say to you is just see, I would just have, like, if you're going to submit it as a separate file, maybe I would put like I would just put in separate file attachment. Just so I know to look someplace else. So if you feel more comfortable, some of you guys may have the skills to be able to actually have a JPEG file or some sort of a GIF or whatever to be able to actually copy and paste it right into the Word document. If you can do that, that's awesome. But if you don't know how to do that, you can attach it as a separate, just take a picture of your graph that you've done and then send it as a separate picture file. And then you'll put a summary. So this is in words, kind of your observations that you made. You should also summarize any trends that you see here. So if you see any kind of trends in the graph that you made, you would summarize those as well. And then finally, your conclusion section is divided up into claim, evidence, and reasoning. So again, you're just restating your hypothesis under the claim. This is the claim that I made. This is the evidence that I collected. So here you're going to go through your data, and your data should, pull out some examples of your data to either support your claim or refute your claim. You're not getting points off, and you're not wrong if you're wrong. If your claim is not supported, then say so here and give evidence that shows it's not supported. And then in the reasoning, you're going to explain why the data that you pulled out does not support your claim or does support your claim. Okay, so that is it. You'll save this, and this will be submitted to me in the Dropbox. Again, if you have any questions with how to set up your tables, or if you feel like maybe you're not 100% sure which experiment you need to do, or you're a little bit unclear, just send me an email. We can make an appointment in my live lesson room, and we can kind of go through this together as well. Otherwise, that's it for this one, ladies and gentlemen, and we'll see you in the next.